GENEie works on the simple logic that annotated human splice sites are known to work, whereas splice site substitutions causing pathogenic missplicing in rare disorders are known to not work. Similar impact and same sequence variants interrogate these simple splicing rules. Rule 6. Known variants at a given splice site can inform the behaviour of a new test variant at the same splice site. Rule 7. Sequence substitutions that disrupt splicing at one intron will likely disrupt splicing at other introns. Rule 8. Sequence substitutions that splice normally at one intron will likely maintain normal splicing at other introns. Similar impact variants are those affecting the same splice site in the same gene as the test variant that elicit highly similar NIF impact across the whole splice site region. We have undertaken comprehensive precision recall analyses to optimize the mathematical parameters which define similar impact. GENEie's similar impact measure understands that not all splice sites are the same. Different splice sites have different thresholds of NIF impact, causing pathogenic levels of missplicing. Let's look at the same splice site variance table for this NF1 plus 5 donor variant in intron 37. GENEie identifies 7 uniquely classified variants in the reference set affecting the same NF1 intron 37 donor. This table separates variants into those with similar impact and those with different impact. The columns show each variant, their position, the NIF impact to each zone, the classification, and a link that directs you to the original submission. Notice all 5 similar impact variants are classified likely pathogenic or pathogenic, and the 2 variants with different impact are classified likely benign. Our statistical analyses show that if your test variant has 1 or more similar impact variants classified pathogenic, this provides 100 to 1 odds that your variant will also cause pathogenic missplicing. Equally, if your test variant has 1 or more similar impact variants classified benign, odds are 100 to 1 your variant will maintain benign normal splicing. So, this NF1 variant with 5 pathogenic similar impact variants is extremely likely to also elicit pathogenic missplicing. Same sequence variants are those making the same substitution in the same 9 nucleotide sequence, but at a different splice site. Let's use this PRKCG intron 13 plus 2 variant as an example. This variant has no similar impact variants reported in the same splice site. This same sequence variants table shows the zone, the window, the reference and variant sequence, and the NIF impact from the NIF table. The right columns tell you that for the E4 to D5 window, this exact same plus 2 T to C substitution within this identical 9 nucleotide sequence has been classified as pathogenic in 5 different introns, supported by 10 submissions, and never classified as benign. For the E3 to D6 window, this exact same plus 2 substitution has been classified as pathogenic for 17 different introns, supported by 35 submissions, and never classified as benign. And so on for every window. Though a 9 nucleotide window comprises the bulk of the splice site, a peripheral nucleotide can alter NIF scores slightly. Therefore, we've defined thresholds that restrict the return of same sequence variants to only those with highly similar reference and variant NIF percentile scores to your variant across the entire extended splice site. This variant GC donor is statistically very unlikely to work. This exact same sequence substitution has occurred in every window across 35 separate introns, and in every case caused pathogenic missplicing.